Hello, my name is Joshua Mugabe. I welcome you to Coordination and Control. Question 6 to 10. Flowering in cochlear plant occur as a result of exposure to a period of darkness. The development of flower has been divided into 8 stages. 0, no flower, up to 8 fully developed flowers. The graph below shows the average stage of flower development for 2 bunches, each of 10 plants, one of which was kept in the darkness for 16 hours and the other for 10 hours. Part A. Compare the effect of length of dark period on flowering of cochlear plant. We shall first look at the similarities. In both bunches, stages of flower development increase with days after the dark period. So if we look here, the x-axis, we have the days after the dark period, and on the y-axis, we have the stage of flower development. So we are saying in both bunches, stages of flower development increase with days after dark period. So you can see that days after dark periods, there is an increase in the flower development for those plants that were kept for 16 hours dark period. And also when you look at those that were kept for 10 hours in dark period, you can also see that there is flower development increment with the days after darkness. In both, less than 2 days of dark period cause no flower development. So we can see that less than 2 days, if they are less than 2 days of the dark period, there is no flower development. No flower development in less than 2 days. So it has to be at least more than 2 days for the flower development to take place. Differences. Let's now look at the differences. Bunch at 10-hour dark period does not reach full flower development after 8 days. Whereas at 16-hour darkness period reaches to full flower development. So those branches that were kept for 10 hours, by the 8th day, they have not yet reached to full flower development. You can see by the day of 8th, they have reached a stage of about 4. Whereas if you look at those ones that are kept at 16-hour of darkness period, at the stage of 8 hours, they have reached the full flower development. You can see they have reached the full flower development. Flower development increases faster and rapidly for the bunch of the 18-hour dark period, whereas the one at 10-hour period, it increases gradually. So the one at 16 hours, the flower development increases very rapidly. Whereas if you look at that one at the 10-hour period, the increase is gradual. It increases gradually. The bunch at 10-hour dark period, it requires more 3.5 days to start flower development, while the one at 6 hours requires only 2 days to start flowering. So if we look at the one of 10-hour dark periods, it requires it more 3.5 days to start the flower development. Whereas if you look at that one kept at the 16-hour, for it, it requires only 2 days to start flowering. Part B. Explain the differences between the two bunches of plants after 80 days. Cochlear is a short-day plant. 16-hour dark period was longer than the critical length, allowing the accumulation of more phytochrome red. Sufficient phytochrome for red is converted to inactive phytochrome red, flowering to occur. Part C. Suggest reasons why there was delay between exposure to darkness and start of flower development. Accumulated growth promoter, those are phytochrome, requires some time to synthesize or to stimulate movement of florigen hormone from the leaves where they are synthesized to the flower bulbs where they exert their effects. Number 7. Distinguish between the resting potential and the action potential. Part B. Explain how the following processes are involved in the formation of nerve impulses. Formation of a resting potential, formation of the action potential, and how the impulse is transmitted. Distinguish between the resting potential and the action potential. Resting potential is a change in the membrane voltage or potential that occur in the nerve muscle or any other excitable tissue in the absence of an excitation. That is, there is no impulse in transit. While an action potential is the change in the membrane voltage that occur in a nerve, a muscle, or an excitable tissue in the presence of an excitation or impulse in transit. Part B. Explain how the following processes are involved in the formation of the nerve impulse. Number 1. Formation of a resting potential. The resting potential is maintained by sodium-potassium pump mechanism and differential permeability of the axon membrane, sodium and potassium ions. At rest, the axon membrane is more permeable to potassium ions and is highly abundant in the oxytoplasm, but almost impermeable to sodium ions, abundant in the extracellular fluid. Therefore, at rest, potassium ion protein gated channels are open, while sodium protein gated channels are closed. Thus, potassium freely diffuses out of the exoplasm to the extracellular fluid without reciprocal entrance of sodium ions. In addition, sodium-potassium pump operates pumping out 3 sodium ions in exchange for the 2 potassium ions. The net effect is a high cationic concentration on the outer axonal membrane relative to the inner, making the membrane positively charged on the outside relative to the inside. This potential difference across the membrane measures up to approximately negative 80 millivolts, and the membrane is said to be polarized. Explain how the following processes are involved in the formation of nerve impulses. Number 2, formation of the action potential and how the impulse is transmitted. Upon stimulation, the sodium-potassium pump breaks down. Axonal membrane reverses the permeability to sodium ions and potassium, in which it becomes more permeable to sodium ions but less permeable to potassium ions. The sodium channels open and the potassium channels close, causing a rapid influx of sodium ions along the steep electrochemical gradient. And this process proceeds by positive feedback. So when you look here we have the resting potential, whereby we have a lot of sodium ions outside. This makes the outside more positive and it makes the inside more negative. So there we say that the membrane is polarized. But when there is an impulse, that means that the sodium-potassium gate are going to open and the membrane will become more permeable to sodium, allowing more sodium to enter inside. Whereas the membrane protein channel in response to potassium ions will close and preventing further outlet of the potassium ions outside. This is going to cause the inside of the axon to become positively charged and the outside to become negatively charged. There we say the membrane has been depolarized. The net effect is the progressively increasing cationic concentration on the inner axonal membrane relative to the outer. The inner membrane, therefore, at rest, potassium protein-gated channels are open, that is at rest, while the sodium protein-gated channels are closed. Thus, potassium ions freely diffuse out of the axon plasm to the exterior cellular fluid, without reciprocal entries of sodium. So that one is taking place at the resting potential. The potassium gates are open and allow potassium ions to go out, but the sodium protein gate channels are closed, not allowing sodium to enter, thereby making the outside to become more positive and inside to become negative. In addition, the sodium-potassium pump operates, pumping out sodium ions in exchange of 2 potassium ions. So still during the resting potential, this polarity is maintained by pumping out sodium ions. Sodium ions are pumped out by the sodium-potassium pump, and potassium ions are pumped inside. For every 3 sodium ions pumped outside, 2 potassium ions are pumped inside. And this results in the inside of the membrane becoming more negative and outside becoming more positive. That is at the resting potential. The net effect of high cationic concentration on the outer axon membrane relative to the inner, making the membrane positively charged on the outside relative to the inside. This potential difference across the membrane measures up to approximately negative 80 millivoltage, and the membrane is said to be polarized. So when the outside is more positively charged than the inside, that potential difference across the membrane measures up to approximately negative 80 millivoltage, and the membrane is said to be polarized, and that's what we call the resting potential. Number 8. Describe the events that lead to perception of a clear image from far object by human during bright light. And part B, discuss the significance of coordinating system of animals. Part A, describe the events that lead to perception of a clear image from a far object by humans during the bright light. Parallel light rays from the distant object will enter the eye through the cornea. It will be refracted through the pupil. The circular muscles of the iris contract. The radial muscles will relax, and limited amount of light is permitted through the constricted pupil. With the ciliary muscles relaxed, the suspensory ligaments are tautened. The edges of the lens are pulled, and the lens becomes thin and less convex, therefore reducing its refractive power. Permitting focus of the parallel rays onto the retina. Light gets incident onto the photopsin or the conopsin, iodopsin, in the outer segment of the cones, and causes the isomerization of the 11-cis retinal to all transretinal, coupled with a conformation change in the protein breaching results. So when the light rays come and strike the photopsin, iodopsin, in the outer segment of the cone, it is going to cause the 11-cis retinal to change to all trans retinal, the one that we are seeing here. We can see this is the cis retinal, and after light hitting the photopsin, the cis retinal is going to be changed into the transverse retinal. And with the conformation change in the protein, breaching results. So what is going to happen in the breaching process? Our rhodopsin and now the transverse retinal are going to separate, and we are going to remain with the ascotopsin and the transverse retin outside, which we call bleaching. This closes the sodium channels and reducing sodium conductance in the outer segment. The membrane of the outer segment gets hyperpolarized. No neurotransmitter release at the inhibitory synapse adjoining the bipolar neuron get depolarized. Release an excitatory neurotransmitter at the excitatory synapse with the bipolar neuron. So because of the light hitting, the sodium channel reduce the conductance of sodium ions in the outer membrane or segment, and the membrane segment becomes hyperpolarized. It becomes hyperpolarized. So no neurotransmitter released at the inhibitory synapse. And adjoining the bipolar neuron, this one gets depolarized, releases an excitatory neurotransmitter at the excitatory synapse with the bipolar neuron. A generator potential established builds up a threshold and creates an action potential that fires an impulse through the optic nerves fiber to the occipital lobe, and this one interprets the image in terms of shape, color, and distance from the eye. So when the generator potential is created because of the built-up threshold, it's going to create an action potential which will fire the impulse through the optical nerve fibers to the occipital part of the brain, which will interpret the images in terms of the shape, color, and distance from the eye. Part B. Discuss the significance of coordinating system of animals. The coordinating system ensures effective cellular communication, that is in multicellular organism, through cell signaling mechanism such as hormonal and neural communication. It ensures optimum cellular conditions, since cellular activities are predominantly enzyme controlled. It enables the organs to overcome stressful changes in the external environment through modifying the physiology or behavior to maintain activity and survival of the organism. It ensures detection of toxicity within the cell internal milieu environment, prompts immediate removal to maintain effective cellular activity. Effective homeostatic regulation of several physiological processes such as temperature regulation, blood sugar regulation, electrolyte balance, and hormonal regulation. Effective reception to stimuli, interaction and appropriate response of the organism to the ever-changing environmental stimuli. Number 9. Describe the mechanism of control of light entering the mammalian eye. Part B. How do photoreceptor cells in the retina of the mammal differ both functionally and structurally? Part C. How is coordination carried out by mammalian nervous system. Describe the mechanism control of light entering the mammalian eye. Much light entering the eye evokes the parasympathetic effect discharge, cause the contraction of the circular or constricted pupillar muscles, and relaxation of the radiodilator pupillar muscles of the eyelids. Papillary constriction results. Limited light is permitted into the eye. So what is happening is that the circular muscles are going to contract, and when they contract, they move towards the pupil, and when they move towards the pupil, that means that the radio muscles have to relax. These radio muscles, when they relax, they will allow the circular muscle to reduce the eyelids. And when they reduce the eyelids, the pupil will reduce in size. And that one will limit the amount of light entering the eye. Limited light entering the eye provokes the sympathetic effect discharge, which causes the contraction of the dilator pupil radio muscles of the eyelids and relaxation of the circular muscle constrictor papilla. Papillary dilation occurs, and thereby permitting more light to enter the eye. Now, in a situation whereby there is little amount of light entering the eye, the sympathetic effect discharge stimulates the radio muscles to contract. These radio muscles will contract, and when they contract, they are going to pull the circular muscles outwards. And when the radio muscles are contracting, the circular muscles have to relax. And when they relax, they will be pulled outwards. And when that one happens, the iris or the pupil is going to widen, or it will increase in the diameter. And when it increases in the diameter, therefore, a lot of light is going to be allowed to enter into the eye. Part B. How do the photoreceptor cells in the retina of the mammals differ both functionally and structure. The photoreceptors, we have the rods and the cones. Now, the outer segment of the rod is cylindrical or rod-shaped, whereas the outer segment is flask or cone-shaped. That one is in the cones. There are more numbers of rods in the retina, about 109 million. And there are fewer in the retina cones, around 106 million. The rods, they contain the rhodopsin as a photosensitive pigment, and the cones contains iodopsin as the photosensitive pigment. The rod demonstrates retinal convergence, whereas in the cone there is no retinal convergence. In the rods, they are incapable of color perception, whereas the cones are capable of color perception. The rod sensitivity to low light intensity, and the cones are sensitive to high light intensity. The rods, they are distributed mainly to the periphery of the retina, apart from the fovea, whereas the cones are dispersed throughout the retina, but more concentrated in the fovea. The rods need lower threshold value, whereas the cones, they need a higher threshold value. Rods increase sensitivity to low light intensity, whereas in the cones, increase visual acuity of the eye. Rods, we have one type, and cones, there are 3 subtypes. Part C. How is coordination carried out by mammalian nervous system? We can see that there is an external stimuli. This one is going to be recognized by the receptors or the sense organism. And these ones will generate the impulse in the nervous system, whereby the coordination response by the brain, and then the information, the response will be taken to the effector organs. And the effector organs that we have are the muscles and the glands. These ones will effect a response. And when a response has been effected, then we are going to get negative feedback to prevent any further stimulation of the receptor sense organs. Number 10. Describe the functioning of hormones in animals. Part B. How is the secretion of aldosterone controlled? Describe the functioning of hormones in mammals. Hormones function by activating signal transduction pathways, thereby influencing physiological, biochemical, and metabolic reactions within the cell. Hormones at the molecular level do so in 4 ways: affecting transcription of genetic information, for example, estrogen; protein synthesis, for example, the growth hormone; altering enzyme activities. We have the example of adrenaline; and changing of the permeability of the cell membrane, for example, insulin. Hormone functioning at the level of the plasma membrane or functioning of the peptide bonds. Amino acids derived from hormones, for example, amines, peptides, polypeptide hormones, being water soluble, they bind to specific receptors on the plasma membrane, transduce intracellular signals by the cyclic adenosine monophosphate, a second messenger system. It begins with the hormone receptor complex being formed, activating adenyl cyclase, which mobilizes ATP molecules, converts them to CAMP, which converts inactive protein kinases to active one. The active protein kinases then phosphorylates several proteins whose effects may alteration of protein synthesis machinery like growth hormones. For example, we have adrenaline here. It will come to the cell membrane, where we have our receptor protein to excite the adenyl cyclase, which will mobilize ATP, convert it to cyclic AMP and 2-phosphate ions. This one will convert the inactive protein kinase to the active one, which active protein kinase. And this one now will phosphorylate the inactive phosphorylase to active phosphorylase, which is going to convert glycogen to glucose, and there will be release of glucose. Alteration of the plasma membrane permits permeability to water and electrolytes, for example, insulin and ADH. It affects the enzyme activity, like for example, adrenaline, or activate the gene transcription, resulting in an increased expression of target protein, like for example, thyroxine. Hormonal functioning at the level of the gene, functioning of steroid hormones. The steroid hormones such as androgen, progesterone, estrogen, and many others, they are lipid soluble and therefore readily diffuse through the plasma membrane of the cells. They bind to specific receptor proteins in either the cytoplasm or the nucleus. If a steroid binds to a receptor in the cytoplasm, the hormone receptor complex moves into the nucleus. The hormone receptor complex influences the protein synthesis machine by binding to the specific region of the DNA, thereby stimulating the production of messenger RNA. That is then translated into specific protein. Part B. How is the secretion of aldosterone controlled? Aldosterone secretion is controlled by renin-angiotensin system, RAS, via the negative feedback. Decreased plasma sodium ions concentration or decreased blood pressure, they trigger the release of renin from the juxtaglomerular complex cells in the nephron. Renin converts inactive angiotensin gain to angiotensin 1, which is also converted to angiotensin 2 by angiotensin, converting the enzymes in the lungs. Angiotensin 2 stimulates aldosterone release from the zona glomerulus of the adrenal cortex. The hormone causes the sodium ion retention and simultaneously loss of potassium ions. Sodium ions are followed by water. If the blood pressure exceeds normal, too much sodium are retained. Renin release ceases and RAS is inactivated. Sodium ions are lost in the renal tubular fluid, followed by diuresis. Potassium ions are then retained. Thank you very much for participating in coordination and control, question 6 to 10. Please do like, share, and subscribe.